Good day to you. Today I'm here to discuss food safety, third semester, second year, where I'm going to talk about Unit 2, Microorganisms in the Food. Talk about microorganisms. These are the tiny organisms which are not visible to our naked eyes, but visible only through microscope. Talk about these microorganisms or microbes. They are categorized into bacteria, virus, fungus, which is further classified into yeast, mold, and mushroom, algae, protozoans, helminths. We'll be talking about this in detail today. Talking about the shapes and structures of microorganisms. As you can see, the moment you say bacteria, you can find in various shapes and structures, fungus, protozoans, and algae. Talk about bacteria. Bacteria is unicellular, single-celled, smaller in size, and it contains various shapes, which I'm going to show you in the next slide, like rod shape, spherical shape, comma, as well as spiral shape. They can form clusters like Diplococcus or can form chain like grapes. They have endospore, which protects them during cooking at high temperature. And the reproduction is by binary fission. As you can see in the slide, for every 20 minutes, bacteria keeps on multiplying. One bacteria enters, after 20 minutes, one bacteria becomes 2. 20 minutes, 2 bacteria become 4. 4 becomes 8, 16, 32. So every 20 minutes, bacteria keeps on multiplying. You can see the shapes of bacteria: circular shape, rod shape, curved other shapes. As you can see, they can form like a rod shape, like a chain, like a cluster of grapes, etc. Talk about bacteria in detail. We need to know what are the conditions that are required for the growth of bacteria into the food. The conditions that are required is factor, food, acidic condition, time, temperature, and oxygen. Food, perishable, semi-perishable, non-perishable. As discussed in Unit 3, food contamination and spoilage, we have been talking about perishable, semi-perishable, non-perishable. Acidic condition, pH should be acidic, so that bacteria will not remain in the product, cannot survive in the product. Time. Generally, the bacteria in the food, it reaches to danger zone within 4 hours. What is the danger zone temperature? 4 to 63 degrees Celsius or 40 to 140 degrees Fahrenheit. Oxygen. Generally, oxygen, aerobic, anaerobic, facultative anaerobes. Aerobic, which survives, these bacteria survives in the presence of oxygen. Anaerobic, these bacteria survives in the absence of oxygen. Facultative anaerobes, they survive either in presence or absence of oxygen. The last one is M, which is said to be moisture. Whenever there is one droplet of water entering into the food, specifically the non-perishable food like pickle, etc., it leads to growth of fungus in the product. So bacteria checks with what kind of food it is, checks the acidic condition, checks the time it can multiply, the temperature at which it can multiply, and checks for the oxygen and moisture. So this is said to be factor: food, acidity, time, temperature, oxygen, and moisture. Coming to the next is growth phase. After the bacteria enters, what is the next step? It leads to 4 phases. Lag phase, which is the first phase, and then there is no multiplication. Log phase or exponential phase, where the bacteria multiplies for every 20 seconds. Stationary phase, where lag phase almost becomes equal to the death phase. Where death phase, the last 2 or 3 bacteria which are left, where the number decreases, they enter into new product and starts their survival. There are some bacteria which is known as psychotropic, which grows at refrigeration temperature less than 5 degrees Celsius. Mesophilic bacteria, which grows at 35 to 55 degrees Celsius. Thermophilic bacteria, which grows at high temperature, that is 55 degrees Celsius and above. This is the graphical presentation, where you can see the graph drawn between the number of population doubling and the log, that is cell density. You can see lag phase, then you can see exponential phase where the graph rises to the top because of the multiplication of bacteria. Then stationary phase, which almost becomes equal to the lag phase. And last, the death phase, again you can see the graph declining because number of bacteria decreases. So we have done with bacteria, where we have been talking about the shapes and structures of bacteria, the shape and structure of bacteria. Then we have been talking about the fathom and growth phase. Next is virus. Virus, these are smaller than microbes. They don't have cellular structure. They have an outer protein layer known as capsid, which wraps the genetic material. They require a host. Host can be either a plant, animal, or a human being. So they are termed as obligate intracellular parasites. They attach themselves to the host and release DNA or RNA. The virus replicates inside the cell, and the cell bursts and the new viruses are released into the atmosphere. So you can see the structure of virus. So the outer layer is capsid, and you can see the internal layer where you have, it wraps the genetic material. And when the host attach themselves to and then releases DNA and RNA, the viruses replicate inside the cell and the cell bursts and new viruses are released. So this is the structure. Next, number 3 is fungus. Fungus is classified into yeast, mold, and mushroom. Talk about yeast. Yeast is single cell. They're spherical or elongated in shape. As you can see here, yeast. This is the yeast. Single cell, unicellular, spherical or elongated in shape. Larger than bacteria. The cell walls have scars known as bud scars. So these are the scars known as bud scars. Reproduction is by budding. The cell forms between the bud and the parent. The bud then separates from the parent and forms into new cell. So you can see here, these are the bud scars which separates from the parent cells and form into a new cell. They are found in dry fruits, fruit juices, jams, and honey. So this is about the yeast. Next is the mold. The entire structure is known as mycelium. The entire structure is known as mycelium. It has got a thin hyphae kind of things, which is said to be a thread-like. This has got a spores, spongy heads, as you can see here in the picture, spongy heads, which is several mm in length and diameter. This spongy head is of different colors, shapes, and structures. So, thread-like called hyphae, spores head, which is 2 to 10 mm in diameter and several mm in length. Several colors like white, black, green, brown, etc. Texture is spongy, powdery, velvety, and these are multicellular. They live in both plants and animals. Advantage, helps in ripening of the cheese. Harmful as it produces aspergillus. Specifically, which you find this aspergillus growth on peanuts. And they are found in bread as well as pickles. When consumed, causes damage in kidneys. They are aerobic. So you can see mycelium. This is the spongy head, 2 to 10 mm in length, several mm in diameter. This is of various colors, textures like powdery, soft, cottony, etc. Next is protozoans. These are unicellular, parasite, and requires hosts. They will form cysts and survive adverse environment condition. As in the cyst stage, protozoan is ingested through contaminated food. So this protozoan enters into human body through contaminated food and water, and they are more organized than bacteria. Help with this. These are multicellular. larger than protozoans. They are microscopic in size. In other stages, they become macroscopic. So starting from eggs to larval stage to adult stage, it becomes macroscopic. It has got a complex life cycle. So these are the various microorganisms which we have discussed today. Bacteria, where we have been talking about shape, size, structure, fathom and growth phase. Then we have been talking about virus, where I have been talking about the capsid, talking about the DNA, RNA, and it requires those to survive. Then we have been talking about the fungus, which we talk about yeast and mold. Yeast, it has got butterscotch which separate from the parents and then develops into new cell, whereas yeast, it has got a thread-like hyphae where it's spongy, soft, cottony with different colors and found in peanuts known as aspergillus and helps in ripening the cheese. Then we have been talking about protozoans and helminthes, where helminthes, it is, you can see it becomes microscopic to macroscopic, starting from egg to larval stage to adult stage, whereas protozoans are more organized than helminthes and they enter into the body through contaminated food and water and then develops inside the body of the human being. So we have done with this. Now, next is the parasites. This stage, protozoans are ingested through contaminated food. Active illness stage is known as active feeding. So these parasite enters through various ways through contaminated food and water, like protozoans, nematode, fluke, hookworm, roundworm, tapeworm. Specifically enters through consuming contaminated pork, which forms round tapeworm, and finally that goes inside the stomach. So they are said to be parasitic. With this, we have completed our unit 2, where we will talk about bacteria, virus, fungus, protozoans, helminthes, and parasites. With this, we have completed this session. Hope you have enjoyed this. Thank you and have a great day.